Hi everyone, my name is Kayal. I'm from the Roy Lab at University of California, San Diego. Today I'm going to talk about the unexpected role of disease-associated serine 129 phosphorylation of α-Synuclein and how this actually is a trigger for normal physiological synaptic function. So α-Synuclein is infamously known for its presence in Lewy bodies, which is a hallmark of synucleinopathies. In neurons under physiological conditions, α-Synuclein exists in two forms. One is a cytosolic native unfolded monomer, whereas the other is the membrane-bound α-Synuclein, which forms alpha helical structures. Under pathological conditions, the monomeric α-Synuclein undergoes major structural changes, causing it to misfold and form oligomers, which then organize into fibrils and then eventually deposit as Lewy bodies inside neurons. And since α-Synuclein is an ubiquitous part of Lewy bodies, diseases which are characterized by the presence of Lewy bodies are called synucleinopathies. And they are Parkinson's disease, dementia with Lewy bodies, and multiple system atrophy. And here is an image of a diseased brain showing deposition of Lewy bodies which are stained with α-Synuclein. So Lewy bodies can be large cytosolic aggregates or they can be neuritic threads. In the case of aggregates, they are called, if they are in cell bodies, they are called Lewy bodies, and if they're in neurites, they are called Lewy neurites. This slide shows an image of the coronal section of the brain, and on the left you have the normal brain, on the right you have a diseased brain, and marked in dark brown are the regions where Lewy bodies are deposited in diseased brains. And Lewy bodies, fine structural analysis of Lewy bodies in postmortem patients reveal that this is a proteinaceous mass. Some can even include vesicular structures and dysmorphic organelles. People have shown that among the proteinaceous structures, α-Synuclein is most abundant in, and it also undergoes many post-translational modifications, such as phosphorylated synuclein and ubiquitin alpha-synuclein as shown on the right. But out of these, phosphorylated synuclein at the 129 site is the most abundant, and this is a hallmark of pathology. And interestingly, this is so abundant that in the case of diseased brains, about 90% of alpha-synuclein is phosphorylated in the serine 129 site, whereas in the normal brain, only 4% of alpha-synuclein is phosphorylated. Because of the stark difference in normal versus diseased brain, studies have always focused on the pathological role of serine 129 phosphorylation. However, a tight regulation of alpha-synuclein in normal brains suggests the physiological function. In fact, numerous examples exist in biology which are regulated by low-frequency phosphorylation events. In fact, initial studies which mapped alpha-synuclein phosphorylation found that alpha-synuclein is constitutively phosphorylated. So here is an experiment that they did where they expressed flag-tagged alpha-synuclein in PC12 cells and treated them with the kinase activators such as forskolin or PDBU, or phosphatase inhibitors such as oxadoic acid. So we can see that by blocking dephosphorylation, they were able to detect phosphorylated alpha-synuclein, suggesting that it is sensitive to phosphatases. So this could be the reason for very low abundance and detection of phosphorylated alpha-synuclein in normal brains. Next, to identify phosphorylated residues, they looked at the structure of alpha-synuclein. So alpha-synuclein is a very small protein, only 140 amino acids, and the N-terminus forms alpha helices, whereas the C-terminus containing the acidic domain is mostly free and unfolded. And they identified two serine residues in the C-terminus, which can be phosphorylated, out of which S129 site was most efficiently phosphorylated, and in fact S129 site is more highly conserved in mammals. So now that So now that we know that α-Synuclein phosphorylation is constitutive and it exists in normal brain, where is it localized? We know that in disease states, both α-Synuclein and phosphorylated synuclein accumulate in Lewy bodies. What happens in normal brains? So α-Synuclein was first identified as a nuclear protein, but later was localized to presynaptic terminals, and hence the name α-Synuclein. And here is an image of cultured hippocampal neurons from synuclein GFP expressing mice. So you can see that they have very faint signal in the nucleus, but predominantly you see punctate structures all over, which are presynaptic terminals. Next, we looked at phosphosynuclein localization by probing for specifically the 129 site detecting antibody, and we see that it localizes in nucleus and also as puncta. To confirm that this punctate structure is actually presynaptic terminals, we did co-staining with VAM2, another presynaptic antibody, and the zoomed image shown at the bottom shows very good co-localization between phosphosynuclein and VAM2, showing that both alpha-synuclein, also that phosphorylated S129P phosphorylated alpha-synuclein localizes at the synapse. So now that we know that both the alpha-synuclein and phosphosynuclein are localized at the synapse, and we know that alpha-synuclein functions at the synapse, and we and others have shown that the abundance of alpha-synuclein at the synapse directly correlates to its function. So is this specific phosphorylation event in alpha-synuclein alter its abundance at the synapse? So to assay presynaptic targeting, we did the following assay. So we co-transfected both alpha-synuclein, GFP-tagged alpha-synuclein and soluble DS-red in cultured mouse hippocampal neurons, and we chose alpha-synuclein knockout mice to prevent any confounding factors. Then we looked at fluorescence. We looked for fluorescent enrichment of the GFP signal in the bouton versus its neighboring axonal regions, and you can see that GFP alpha-synuclein accumulates in the boutons but not in the neighboring axonal regions, whereas the soluble RES-red, which is a volume filler, is uniform in both the bouton and in the neighboring axonal regions. So below is the formula we used for calculating the targeting factor. So as you can see on the right, soluble GFP, which does not localize, which is not enriched in synapses, has a targeting factor of 0, whereas compared to wild-type alpha-synuclein and the N-terminus of alpha-synuclein, which have a very high targeting factor because they are enriched in synapses. And as expected, the C-terminus of alpha-synuclein cannot localize to synapses because the N-terminus has the alpha helical domains which are required for membrane binding. So we showed that this shows that the N-terminus of alpha-synuclein is required for presynaptic localization. In fact, we also looked at mutants which are affected for membrane binding, such as the familial mutant in Parkinson's disease, the A30P, and also the T6K mutant, which has been designed to disrupt the alpha helical structure. And since both of these cannot target to the synapse, this clearly shows that the N-terminus of alpha-synuclein is required for presynaptic localization. Next, we wanted to test if phosphorylation contributes to presynaptic localization. So as mentioned before, alpha-synuclein has 2 serine residues which can be phosphorylated, but out of these, the S129 site has been shown to be more efficiently constitutively phosphorylated, and also the 129 site is highly conserved in mammals as opposed to the serine 87 site. So to study how phosphorylation contributes, we mutated the serines to either an alanine, which prevents it from being phosphorylated, making it a phosphonull mutant, and we also mutated serine to an aspartate, making a phosphomimetic mutant, which is classically being shown to mimic phosphorylation. So next so does phosphorylation affect the presynaptic targeting of alpha-synuclein? So to do this, we co-transfected the phosphosynuclein mutant constructs and DS-RED and looked at its localization at the presynaptic terminal. And strikingly from the image, it shows that the 129 phosphomimetic mutant, the 129D mutant, is augments synaptic targeting of alpha-synuclein. And we know that this synaptic targeting is specifically due to the 129 site, because if you look at the data from the double mutants, even when the S87 site is a phospho-null mutant, it does not affect synaptic targeting. So this clearly shows that the S129 phosphorylation acts as a switch that augments alpha-synuclein presynaptic abundance. In fact, to test this, we also took the mutants, the A30P and the T6K, which were affected for synaptic targeting, and now we introduced the 129 phosphomimetic mutant in them, and they significantly increased synaptic targeting. So collectively, this data shows that though S129 phosphorylation is not required for synaptic targeting, the N-terminus alone is sufficient. However, dynamic phosphorylation and dephosphorylation events may regulate its binding to synaptic terminals. Now we saw that the increased alpha-synuclein phosphomimetic can increase synaptic targeting, and we know that the levels of alpha-synuclein at the synapse is directly linked to its synaptic recycling function. Is directly linked to its synaptic recycling function. So we and others have shown that alpha-synuclein is an attenuator of synaptic vesicle recycling. So when we express human alpha-synuclein in mouse synaptic neurons, alpha-synuclein clusters synaptic vesicles, prevents mobilization, as a consequence of which synaptic vesicle recycling and neurotransmission are reduced. So now that we know that the alpha-synuclein 129D mutation increases synaptic targeting, we hypothesize that overexpression of this mutant will further attenuate this activity-dependent synaptic vesicle recycling. So to look at synaptic vesicle recycling, we use pH-sensitive fluorescent probes, called V-glued fluorine. V-glued fluorine, when expressed in hippocampal neurons, binds to the inside of synaptic vesicles, and its fluorescence is quenched because of the acidity of the lumen. And you can see the GFP fluorescence profile shown below. And upon receiving a stimulus such as the electrical stimulus, in this case 600 action potentials, vesicles are mobilized to the surface, so exocytosis happens, the pH changes, so there is an increase in fluorescence, a steep increase in fluorescence at step 2. Then this is followed by a gradual decrease in fluorescence when retrieval of the membrane or endocytosis occurs. Finally, we flood the system with ammonium chloride solution, which is alkaline, so it reveals all the entire synaptic vesicle pool. So we can use this to determine the percentage of synaptic vesicles released as a consequence of the electrical stimulus. So here is an example of hippocampal neurons transfected with V-glued fluorine, and the first image shows baseline fluorescence before stimulation. Then you can see the increase in fluorescence at boutons after stimulation, and finally the ammonium chloride, which reveals the entire synaptic pool. So we used this fluorine assay, and we transfected neurons with either the empty vector or wild type alpha-synuclein. And you can see that at 30 seconds is where we gave the 600 action potentials, and you can see the increase in fluorescence in both the empty vector and in the wild type. But in the case of neurons where wild type alpha-synuclein is overexpressed, synaptic vesicle recycling is attenuated when compared to the control. So since the S129D mutation increases synaptic localization, we hypothesize that this overexpression will further attenuate the synaptic vesicle recycling. And in fact, that is what we saw, that the D mutation further reduced synaptic vesicle recycling. So this shows that alpha-synuclein 129 phosphorylation indeed contributes to the normal function of alpha-synuclein. So next we wanted to look at the phospho-null mutant of alpha-synuclein, the 129 null, phospho-null mutant of alpha-synuclein. So recall that this mutant can localize at localized at presynaptic terminals similar to the wild type. So can it also suppress synaptic vesicle recycling similar to the wild type? What we saw was pretty surprising because the S129A mutant can localize to synapses, but it could not suppress synaptic vesicle recycling. So this shows that alpha-synuclein phosphorylation is essential for the normal function of alpha-synuclein. So since this physiological role of, so this alpha-synuclein phosphorylation is required for activity-dependent alpha-synuclein function at the synapse, and to the best of our knowledge, this is the first described unexpected physiological role for alpha-synuclein 129 phosphorylation. And since this physiological role of alpha-synuclein is activity-dependent, we asked if we can increase 129 phosphorylation by increasing neuronal activity. So to do this, we used either chemical activators or electrical stimulus. So first we used 4-AP or 4-aminopyridine, which has been shown to increase neuronal activity by blocking potassium channels, and it has been used in the treatment of several diseases. So first we treated cultured neurons with 4-AP for different time periods, and then we did the Western blot. So as you can see below, the total amount of alpha-synuclein does not change over time when treated with 4-AP, but the amount of phosphorylated alpha-synuclein is highest at 180 minutes after treatment with 4-AP. And here is the quantification showing that, in fact, treatment with a chemical stimulator such as 4-AP increased alpha-synuclein phosphorylation. Next, we wanted to know if electrical stimulation, the same paradigm that we used in our fluorine assays, also can increase phosphorylation. So here are cultured cells stimulated with 600 AP, and in the rightmost panel we see that upon electrical stimulation we increase S129 amount of S129 phosphorylation detected. The second lane shows that this is specific to electrical stimulus because treatment with TTX, which is a sodium channel blocker, then the neurons cannot be activated, and hence we block synuclein phosphorylation. So this clearly shows that inducing activity by electrical stimulation and by chemical stimulation increased alpha-synuclein phosphorylation. So this phosphorylation, increase in phosphorylation, is indeed activity dependent. Now we wanted to know if this increased activity can increase phosphorylation also in vivo. So for this, we injected mice with the peritoneally injected mice with 4-AP and harvested the brain after 3 hours and did biochemical analysis on this. On the left, you have 3 mouse brains injected with PBS controls, and on the right, you have 3 mouse brains injected with 4-AP controls. And we can clearly see that in the 4-AP treated mice, as shown in the quantification, there is an increase in serine 129 phosphorylated alpha-synuclein. So inducing activity both in vitro and in vivo increased alpha-synuclein 129 phosphorylation. So we know now that neuronal activity triggers this phosphorylation, and this phosphorylation is required for function, but the mechanisms are not known. Typically, multiple proteins work together in the process of synaptic vesicle recycling to coordinate snare assembly, vesicle fusion, and endocytosis, to name a few. And such a functional network of proteins is also starting to emerge for alpha-synuclein. So we asked if this alpha-synuclein 129 phosphorylation influences binding of alpha-synuclein to its interaction partners. So the Roy lab has previously shown that alpha-synuclein binds to two presynaptic proteins, VAMP2 and synapsin, and this is required for its function in synaptic vesicle recycling. So very briefly, we already identified that this specific residues, KKD, in the C-terminus of alpha-synuclein binds to VAMP2. So in fluorine assays, we know that overexpressing wild-type synuclein, shown in red, attenuates synaptic vesicle recycling. But now when we express alpha-synuclein in which the KKD is mutated, which is the green line, shows that synaptic vesicle recycling cannot be attenuated, so, showing that both alpha-synuclein and VAMP2 interaction is essential for alpha-synuclein mediated function at the synapse. Next, we looked at alpha-synuclein and synapsin. So again, on the left, we have a fluorine experiment from wild-type neurons showing that overexpressing alpha-synuclein, shown in orange, reduces synaptic vesicle recycling. On the right, we have synapsin triple knockout mice in which synaptic vesicle recycling cannot be suppressed, showing that synapsin is required. Now, in this context, if we again reintroduce synapsin 1 and also overexpress alpha-synuclein, this suppression is restored, suggesting that the interaction between alpha-synuclein and synapsin is also essential for this function in synaptic vesicle recycling. So we showed that alpha-synuclein phosphorylation increases as a result of neuronal activity. So does this increased phosphorylation result in increased binding with both VAMP2 and synapsin? So to do this, we co-transfected neuro2A cells with MIC-tagged alpha-synuclein wild-type or phosphomutants and green lantern-tagged VAMP2. Then we did an IP with the green lantern antibody to pull down VAMP2 and then detected alpha-synuclein using the MIC antibody. So on the image here, you can see that the fourth lane shows interaction between wild-type alpha-synuclein and green lantern VAMP2, whereas the adjacent lane where the S129A mutant is co-transfected, we do not see any interaction with VAMP2. Whereas in the case of the phosphomimitic S129D, we see an increased interaction of alpha-synuclein and VAMP2. And a similar pattern on the right, we did the same IP, now expressing green lantern tagged synapsin, and we see a similar profile where mutating the S129 site to a phospho-null mutant completely abolished the interaction between synapsin and VAMP2. So this shows that phosphorylation of alpha-synuclein at the single residue as the one at the S129 site is required for binding. And because just by mutating this one residue, we can completely disrupt binding of alpha-synuclein to both synapsin and VAMP2. So the previous experiment was done in vitro in cell lines. So we next asked if this interaction occurs in the context of the brain. So to do this, we took mouse brain lysates and incubated them with in vitro purified GST-bound wild-type synuclein and phosphomutants. Then we did a pull down with GST and identified proteins which bind to alpha-synuclein. So on the second lane shows that the interaction occurs between wild-type alpha-synuclein, and we can pull down both VAMP2 and synapsin, and this is specific to alpha-synuclein because the GST controls shows nothing. And strikingly, again, when the S129 site is mutated to an A, preventing it from phosphorylation, the binding to both VAMP2 and synapsin is abolished. And the phosphomimetic mutant can still interact with both VAMP2 and synapsin. And here is the quantification on the right. So this clearly shows that the α-Synuclein phosphorylation binding to VAMP2 and synapsin occurs endogenously in mouse brains. So if this interaction is real, the S129 α-Synuclein phosphodependent interaction, does it occur in vivo, and is this activity dependent? So to do this, we injected mice with 4-AP, and 3 hours later harvested the brains and did an IP with VAMP2 or synapsin and looked for the presence of phosphorylated α-Synuclein. So the blot shows that in the last lane where VAMP2 is pulled down and the mice are treated with 4-AP, we see an increase in phosphorylated α-Synuclein as compared to the PBS treated control. And a similar profile also, we see a similar profile when also 4-AP treated mice are pulled down with synapsin. We see an increase in phosphorylated α-Synuclein. So this clearly shows that the S129 α-Synuclein phosphodependent interaction is a consequence of neuronal activity, and it is a consequence of neuronal activity that is occurring in vivo. So a final summary, we showed that this activity-dependent phosphor switch at the α-Synuclein 129 site triggers association with its functional interacting partners VAMP2 and synapsin. So now the question is, how does the single phosphorylation event modulate α-Synuclein interactions? So we know that while the N-terminus binds to membranes, the C-terminus is free and folded. So to predict the effects of α-Synuclein 129 phosphorylation on structure and membrane binding, we used AlphaFold based modeling. So for this, we compared the unphosphorylated state of α-Synuclein with the phosphorylated state of α-Synuclein, and we saw that in the case of phosphorylated α-Synuclein, more of the residues, C-terminal residues, are available for interaction, whereas in the case of unphosphorylated, up to 97 residues, more of the C-terminus is buried inside the membrane. So in the case of α-Synuclein 129 phosphorylation, it exposes more of the C-terminal residues, suggesting that it is more available to bind to its interaction partners. So in conclusion, we show that the neuronal activity increases α-Synuclein phosphorylation, which leads to increased synaptic targeting and consequently to increased binding to its interaction partners. So in conclusion, we showed that the neuronal activity increases α-Synuclein phosphorylation, which leads to increased synaptic targeting and also increased VAMP2 and synapsin binding, which consequently plays a role in synuclein function. So this is the first physiological role described for this disease-associated serine 129 phosphorylation site at the synapse. So what happens in disease? So the same function could contribute to pathology, or it could be a toxic gain of function, which is still to be explored. Typical multiple proteins work together in the process of synaptic vesicle recycling to coordinate exocytosis, fusion, and also endocytosis. So now we saw that phosphorylation of the single residue in α-Synuclein. So we showed that phosphorylation in the single residue in α-Synuclein regulates its binding to different partners, VAMP2 and synapsin. And we think there could be a much bigger repertoire of proteins that α-Synuclein phosphorylation regulates binding to. So currently we are exploring the different partners which bind to α-Synuclein as a function of its phosphorylation. with that, I would like to give thanks to all the members of the Roy Lab, to my mentor Subhojit Roy, to Leo, who did all the beautiful biochemistry in this project, to Lena, who did the fluorine experiments and targeting, and Nick, who helped us with modeling. And our collaborator, Padmini Rangamani, who helped us with the modeling also as well. Thank you.